Good day, everyone. Today we want to talk about the cyclic AMP, which is also referred to as cyclic adenosine monophosphate. And because of the relevance of this second messenger in our body system, it becomes so relevant to us as biomedical students and also as a researcher and also as a scientist to study the impact of this cyclic AMP as a second messenger in biological processes. And what is the relevance of cyclic AMP to our body system and some cellular functions we have in the body? Cyclic AMP, as it being called, which is also referred to as cyclic 3' 5' monophosphate, as a second messenger. They function in different process in the body system. One of it is immunological function. They also function in memory. And it has also been discovered that they function in transcription process in our body system. And also is that cyclic AMP, they mediate metabolic processes, as we are going to unravel later in the course of our study. Basically, just because cyclic AMP is playing a very significant role in signal transduction, it becomes so important to play a central role in mediating so many biological processes, and some of it you're going to see as a function to be explored for therapeutic goals, as the case may be. Usually or generally, first messenger cannot cross the membrane to the other side of the cells to carry out a signal transductional process. And because of this, external stimuli needs to be converted to internal stimuli by way of cyclic AMP. So cyclic AMP is standing as intermediate between the external stimuli and internal stimuli for signal transductional process. And therefore, work related to cyclic AMP. How does cyclic AMP mediate this function? Every ligand in form of neurotransmitter, prostaglandins, hormones, binds to a particular receptor at the surface of the membrane. And the very receptor we want to talk about today that is very related to cyclic AMP is the 7-transmembrane receptor coupled Gprotein, or what we refer to as Gproteincoupled receptors. Gproteincoupled receptors. So this 7-transmembrane Gproteincoupled receptor is what has been depicted here. This is Gproteincoupled receptors. By action of the ligand binding to Gproteincoupled receptor at the surface of the membrane, they cause one of the subunits referred to as G-alpha subunits. They cause one of the subunits of the Gproteincoupled receptor, G-alpha subunit, to detach away from its other member of the Gproteincoupled receptor, which are beta and gamma subunits. So by so doing, this dissociation of making GDP to be exchanged for GTP will make G-alpha subunit of the G protein to detach away from the beta gamma subunit, which were initially associated together. So by so doing, you alpha subunit now interacts with the enzyme, adenylyl cyclase enzyme. And what is the function of the adenylyl cyclase enzyme? The major function of adenylyl cyclase enzyme is to convert ATP, adenosine triphosphate, into cyclic AMP, right? And the converted cyclic AMP has its own effector molecules. And the two have been identified effector molecules of cyclic AMP is one, protein kinase A, which is referred to as an abbreviation pKa. We also have what we call cyclic nucleotide ion gated channel. And the third aspect is what we refer to as guan GTP nucleotide exchanger. GTP nucleotide exchanger, which could be called EPAC as abbreviation. These are the three major discussed effector molecule of cyclic AMP. When we talk about effector molecule, we're talking about those molecules in which cyclic AMP can bind to as a way of crosstalk to control some other cellular transductional process. All right. So for the case of protein kinase A, protein kinase A as a phosphorylating molecule, binding of cyclic AMP, binding of the cyclic AMP to the subunits of pKa. pKa has two major subunits. The first subunit is what we call regulatory subunits of pKa. The second is what we call the catalytic subunit of pKa, catalytic subunit. So binding of cyclic AMP, cyclic adenosine monophosphate, to the regulatory subunit of pKa, protein kinase, will release the catalytic subunit of the pKa. So the release catalytic unit of the pKa now goes to activate some transcriptional factors, right? They go to activate some transcriptional factors. So basically, this is what we're talking about. We have cyclic AMP, that has been formed from adenosine triphosphates, whereby in the presence of adenylyl cyclase, you have ATP being converted to cyclic AMP by a cyclization process, whereby ATP forms cyclic AMP and pyrophosphate. So we are concerned with the cyclic AMP that is formed. The cyclic AMP formed binds to the subunits of protein kinase A. The subunit of protein kinase A, we say they are divided into two. The first two units, we call it regulatory units. We have this called catalytic unit, catalytic subunits. So binding of the cyclic AMP to the regulatory unit dissociates the catalytic unit of the protein kinase A, whereby this now go to phosphorylate. It goes to phosphorylate the transcriptional factors such as cyclic AMP response element binding protein, and also activate also nuclear factor kappa B, and also the transcriptional factor CREM. So by way of this activation, you can see that cyclic AMP is playing a central signal transduction which controls some transcriptional factors such as cyclic AMP response element binding protein, and also nuclear factor kappa B. And all of these transcriptional factors goes to bind with parts of the DNA promoters. So they are promoting gene transcriptional process. And what is the relevance of this? We've said that among the functions of this cyclic AMP biological process, signal transduction, is that one, the function in immunological process. Two is that the function in memory. Three, metabolic process. Four, gene transcriptional process. Right. So let's identify each of those functions of cyclic AMP based on its relevance to clinical and biomedical practice. Cyclic AMP plays a major role in some of the immunological process we have in the body system. And basically, based on the research that has been done so far, it has been identified that increase in cyclic AMP suppress the expression of immunological processes. Probably need to repeat that again. Cyclic AMP has been associated, increase in cyclic AMP has been associated to suppress the expression of immunological process in our body system. And this can be seen in pathogenic microbial organisms. When a pathogenic microbial organism enters into the cell by way of microbial invasion, one of the methods in which this microbial organism tends to do to evade defensive mechanism by the immunological process is actually to explore the whole cell cyclic AMP. And by way of increasing this whole cell cyclic AMP, they tend to suppress the activity of the immunological functional cells, such as macrophages and the leukocyte, basically. And how it does it is that they suppress the mechanistic function of these immune cells by depressing reactive oxygen species, phagolysis process, and other process of immunological process that are relevant to fighting a pathogenic invasion. So by so doing, they are doing what we call suppression of the immunological cells, as the case may be. All right. So, and importantly, also is that this cyclic AMP also mediates memory cells. Memory cells. How do they mediate the memory cells? It has been discovered that increase in the action of this cyclic AMP, increase in the action that will lead to increased production of cyclic AMP, promotes neuronal cell survival. And as a result of promotion of the neuronal cell survival, it tends to increase the level of the long-time plasticity and potentiation at the level of the central nervous system. And also talking about the metabolic process of the cyclic AMP. Basically, the metabolic process of the cyclic AMP can be seen in the process of insulin release. Increased cyclic AMP bring about increased release of insulin in our body system, and also some neurotransmitters such as adrenergic agonist receptors will also mediate the process of increased cyclic AMP production. And talking about the cyclic AMP function in relation to gene transcriptional process. Basically, the gene transcriptional process of cyclic AMP is mediated via cyclic AMP being binding to protein kinase A subunit, regulatory subunit portion of the pKa. So by binding to the protein kinase A subunit of this molecule, we bring about phosphorylation of the transcriptional factors such as nuclear factor kappa B and also cyclic AMP response element binding protein. All of these go into the DNA, thereby promoting gene transcription, promoting new cells promotion. All right, those are the major functions in which the cyclic AMP participates in to bring about signal process transduction. Also, the regulation of cyclic AMP basically is from 1, adenylate cyclase enzyme, and 2, phosphodiesterase, which is called PDE, phosphodiesterase enzyme. These two enzymes regulate the balance of cyclic AMP at the cellular level. So anything that will cause the production of cyclic AMP will activate adenylate cyclase. Anything that will reduce the formation of cyclic AMP will activate phosphodiesterase enzyme. So phosphodiesterase enzyme naturally degrade the cyclic AMP, right? So that is the process in which cyclic AMP in the body system is being regulated, all right? And it has also been said that majority of the pathogenic, we have said it earlier on, they function by way of exploring the cyclic AMP that we have in the body system. And we have discovered some drugs that actually also uses or utilizes, we have some drug that actually utilizes this cyclic AMP as a way to actually carry out their therapeutic goal when it's been instituted in the body system. So this is the major basic knowledge about cyclic AMP. Cyclic AMP is playing a very critical role and prominent role, so to speak, in signal transduction, biological process, and also it crosstalk with other signal transduction to bring about the execution of a stimuli, which could be in form of a neurotransmitter or hormone being secreted at one point of the organ of the body of our system. So by so doing, we can rightly say cyclic AMP can be explored for therapeutic goal in our system to mediate some biological function by way of modulation, and also to bring about it being reduced in some pathogenic process of the disease condition. And all of this play very important role to talk about how relevant a cyclic AMP is functioning in our body system. All right, so because of our time, I think subsequently I will employ you to join me for subsequent videos. We're going to be talking as to biomedical relevant topics that is necessary to be understood by basic medical students and also clinician, as the case may be. Thank you very much.